Good evening, everybody. Thank you so much for joining this session. So today we will be more discussing about do's and don'ts in QSAR. Before that, I would recommend everybody either on a phone or maybe on other interface. You can go to the website called mentee.com, and it will be asking for a code. You can enter the code 741474. I repeat, 741474. Throughout the meeting, you can ask for questions here. In any slides, you can ask questions. So when you ask questions, you can see that at the bottom of the screen, the questions numbers will be coming. So already I can see 5 questions are being asked by somebody else already, who already knows about Menti, I think. So if someone is new, at the bottom of that page, once it opens, there will be ask question will be there. You can press that ask question and you can type your question so that it automatically come, and during the question and answer session we will take up the questions and discuss. So myself Giri, my PhD research was mostly into QSAR and computational chemistry, and of course we have applied statistical methods and machine learning methods for the QSAR models. So as usual, so please look at this. People who have joined via Google Meet or Hangout Meet, you have to kindly switch off your webcam for better buffering, so that there won't be any issues with audio or video. And you will be in mute mode. You could raise hand option to engage in discussion, or you can unmute yourself during the question answering session if you want to ask any questions. Otherwise, do not unmute yourself. So then kindly tell your name before engaging in any discussions via audio, and questions can be shared only via Menti, or if you think that you are not comfortable, you can always unmute yourself and then go ahead and ask the questions. Agenda of the meeting will be decided based on the participants' interest as usual from the second slide. Now, in your mentee.com, you can start casting your vote. So there are several areas that I have listed here. So if you go to the page now, it will be asking for which area that you want to choose. I think you can choose up to 3 or 4 options. So based on that, we will today take up the questions, and I will discuss about that in the same. So I can see already two people have already submitted it. So let's see how far it goes. Okay, submissions are coming in. So I can see more of machine learning methods, QSAR models. Okay. We have around 29 participants in different. So only 5 people have responded. So this will give me a better understanding where we have to more focus on. So these all are anywhere related to QSAR only. So I can see now more have chosen QSAR models. Yeah, coming in. Okay. I think you can choose 4 options at least. Yeah. By the way, if you have free time, you can always start asking question. And the same page at the bottom, there is ask a question. So you can start typing the question, so that when we have a question and answer session, we will be able to cover them one by one. Okay, so I think that's fair enough. So we have to concentrate more on QSAR models, and then Cheminformatics and others. So okay, that's good. Great. So today we will be more concentrating on QSAR models as well as Cheminformatics. So I will give an overview about how to calculate descriptors, what is its significance, what is the right way to do it. That I will give a briefing through some slides. So, and let me go to the next slide to just understand how far is your confidence on different areas. So again, in mentee.com, you can slide based upon your expertise and understanding. So you can do for all or very few. So if you have like, you can submit it, like you can choose whatever you want. So you just slide or just click on each of the riders or slides and then say what is your maximum, are you an expert in all of them. So you can either give it for all or one, it depends up to you, your choice. Yeah, I can see already 2 people have submitted. Just to better understand where I have to focus more. Okay, so okay. Okay, it's a balance of all. Okay, sorry for the typo. It is Knime, Knime. Somehow I missed it. Sorry for that. Okay. Okay. So you're not used to the combination of that particular tools, which is open source, of course, free for academics, and some in model validation, which is very important in QSAR. And I can see 2D QSAR also a little bit confused there. Okay, great. That's great enough for me to start with. So before going for the next question is and my presentation, today we will be discussing about the topics that rank top, and you already know what are the things that we are going to discuss. All of you should refer to only open source data and no promotions are entertained here. All the opinions and discussions are personal and personal opinion, and it does not relate to with connection of any current employees or corporates or any other individuals. So please keep in mind there is no conflict of interest here. Okay, so so I'm going to the presentation. So when I'm going for these presentations are publicly available. Of course, it is in my SlideShare. So you can go to slideshare.net/giribio, GIRIBIO. There are several presentations available. Of course, in the coming days, I'll be uploading more. Now I'll concentrate into this particular presentation, role of drug design in medicinal chemistry. It is in the 53rd slide. I'm not going to present everything. Part by part, I will be presenting in the coming weeks. So I'm just concentrating on the QSAR, and based on this presentation, we can have a questionnaire session in the coming in the following minutes. So coming to QSAR, we are talking about quantitative, structural, activity relationship. Here, what we are trying to do is, for example, the purpose of QSAR is, let's say you have a diverse set or a homologous set of compounds available with the biological data. Biological data in the sense could be bioassay, or it could be some biological property or even activity. It can be chemical, it can be physical, or it can be biomedical. So per se, I didn't mean that it to be only biologically. For chemist also it is available, for physicist also, or even for material science also it could be done. But since we are doing a structure based quantitative activity relation, we call it as QSAR. Some people call it as QSPR, that is property relationship. Some people call it as QSTR, that is quantitative structural toxicity relationship. Now, the data required for this is a structure, that is a chemical structure, and or smiles. It could be a 2D structure or it could be a 3D structure, but you need a chemical composition understanding or a IUPAC name or inch keys or canonical smiles, whatever format you have. The next thing what you need to have is a experimental data. It could be anything. It could be on any units, but we have to normalize it. We are trying to relate them to understand how far the structural property is dependent on those chemical or physical or biological property. We are trying to relate them, or we are trying to correlate them. This correlation process is being achieved by statistical or machine learning process or methods. For this process only, we altogether call it as QSAR. Now, why we are doing it? The purpose of doing this is in order to predict the activity for new set of molecules. For example, we want to design a new molecule. As a chemist, I don't know anything about biology. So I know better making or designing new molecules. So when I design a new molecule, what could be its possible properties? As you are aware, I know that many of you are using PASS server, PASS server. They also use QSAR based models to understand which of your compound, the compound that you submitted, having which activity among the listed. I think there are more than 20 or 30 properties being listed there, or activity being listed there. So we are trying to achieve a reverse model to understand, okay, for my particular compounds, how the activity could be based on my models. So in order to generate this model, we need to have the structure as well as the chemical structure as well as the biological or experimental activity from a bioassay. Now, how do we generate or how do we correlate them in order to generate the QSAR model? We try to extract, I hope you can see my cursor, we try to extract the numerical values from our molecular structures. They are called as molecular descriptors. And these descriptors can be 2D, two-dimensional or three-dimensional, and there are several types of descriptors which I will be discussing in the next slides. And then I wanted to convert my activity data, also called as response variable, in statistical or in statistics. Now coming to response variable, the problem here is we have a different set of molecules and the activity might not be, what to say, normalized. So in order to normalize the data, we take logarithmic value. So log values we try to take. Log of IC50, we try to take the value to normalize the data. But keep in mind, when you're trying to predict anything for your new molecules, ensure that sorry, okay, I think you are not able to see my slides. Oh, okay, let me check, just a minute. Sorry for that. I hope now you will be able to see my slides. Okay. So this is what I was telling you, the structure and experimental value. From structure, we have extracted the molecular descriptors. Of course, we use some software. By the way, if you are not able to see my slide, kindly let me know so that I can check it out. You can put in the chat message. I hope you are able to see my slides, right? Can you someone respond to me? Okay. Okay, for most of them it is visible, for some it is not visible. So can you try to refresh your Meet, or if you want to see the slides, you can go to YouTube also. The YouTube link is live and there the slides are visible anyway. Sorry for the glitch. Okay, for most of them they are able to see it. That's what I get the response. Thank you so much for that. So the molecular structure, we are extracting the numerical data or categorical data. It could be anything based on the kind of descriptors you are trying to calculate. That is a molecular properties. And then we are trying to relate it with the response variable. Now the response variable could be categorical data or it could be numerical data also. So now we wanted to see how to relate X with Y. So that's what we are trying to do. How these or which of these molecular descriptors could be correlated with this activity. That is a process. So for that process, we do feature selection or descriptor selection. It is usually using genetic algorithm methods. We try to find out which of these descriptors are having more significance to correlate with this activity. And then we use different statistical or machine learning models or methods to try to find out how this can be correlated better. What we are trying to correlate it, whatever the descriptors are having significance to correlate with the response variable or our activity, experimental or wet lab activity, we found those descriptors and we formed an equation saying that activity or property is a function of physiochemical property is a structural property with its coefficient plus or minus error. Very important this, please don't forget to put error value in QSAR models. In many papers, I have seen people are not reporting the error. The error value is very important in statistics as well as in machine learning. That tells you how far your models are reliable. It's not just your R-square is going to say that your models are reliable. No, never. So we need to have a better understanding or a balanced what to say, acceptance of all these statistical parameters. Now, in order to calculate the R-square, what we do is we have the experimental data with us, and based on this models with the descriptors as well as the activity, we have calculated the predicted data also. Now we try to plot a scatter plot and correlate the data to understand how far they are like correlated between the predicted and the experimental data, and we could see that's where you get the R square. So here R square can come to 82 or 90, but don't expect 100. If it is done 100, there is something not right there. Once these models are generated, that is called, now we generated a training model or a training set. After that, we have to validate this model. Is this model is really reproducible? Whether this model is really reliable? That's what we wanted to understand now. So in order to do that process, we do a basic analysis of a validation step. Now the validations are several modes: internal validation, external validation, and we also do manually also. After this validation is set, that's where for your new set of molecules, you can use the formula or the QSAR model equation to predict the property. And that is the process now. That whole together is called QSAR. Now, what we are trying to substitute, or what values that you can achieve or you get from the training set is, you will get the intercept value, the constant value, as well as the coefficient of the significant descriptors from this model that will form this equation. So that when you try to form this equation, and then you will substitute only the descriptor values of the new set of structures. So let's say you designed a new molecule which never synthesized, never tested. You just draw the structure, calculate the descriptors, and you just substitute the specific descriptors that came up in the model, the significant descriptors, and you will be able to predict the activity. But keep in mind, the activity predicted will be log of IC50. So keep that in mind, because we took a logarithmic log of IC50 of our activity for our models. So whatever you are predicting also will be log of IC50. You have to, in Excel, you can take the power of that value in the 10 power, so that you will get the actual IC50 value. So that is where you are trying to predict it. Now let me go through the process how we did this. So you need to have a molecular structure. We have to optimize it, descriptor calculation, classification, feature selection, model generation, and validation. Now let me give a very big disclaimer here. Don't think this can be done in a click of a button. No, QSAR is not a process that you can do in a single click of a button. It is a several process, and every process you have a checkpoint that to be checked and clarified. Please keep that in mind, very important. So I'm going to the next slide. So how to elucidate this molecular structure? The molecular structure can be in IUPAC name or smiles, canonical smiles, or it can be in a 2D structure, and the 2D structure can be converted to 3D. And now I have to also emphasize on 2D descriptors are different, 3D descriptors are different. 2D descriptors takes from 2D structures. 3D descriptors takes the descriptors from 3D structure. Because when you come with 3D, there are quantum chemical descriptors coming into the picture, thermodynamic descriptors coming into the picture. When you go for 2D descriptors, it's more of topological descriptors or constitutional descriptors. So that's where the descriptor values also will be different. Coming to 3D descriptors, you have to be very careful, because unless you have a biological complex conformer of your molecules, most of the time I don't recommend. Otherwise, If let's say you don't have a structure based molecular docking possible for your molecules, which is for QSAR, that's where we do QSAR anyway. So in that case, we have to at least do pharmacophore screening and then come up with the possible conformation which is very close to your reference pharmacophore map, and consider that conformer to calculate the descriptors. This is the reason why you have to be very careful while calculating 3D descriptors. So don't think that clicking a button, just simply calculating 3D descriptors are always the correct one. A change in a small descriptor value has tremendous amount of change in the error, of course, false positives also. So keep in mind that 3D descriptors, if you plan to use, ensure that your starting geometry or the conformer of your compound is either taken from a docked complex structure. That means you do a docking analysis and you made some of course very well interpretations and picked up a pose from there, which is a conformer, and you're going to fix that geometry and try to calculate the descriptor from there. Second, if you don't have a protein based docking study available, then you go for a pharmacophore based. That's where we earlier had a COMFIA or molecular field based analysis or pharmacophore based analysis. So you have a reference molecule. Based on that, you try to generate possible conformations of your molecules and understand what could be a pharmacophore fit conformer. Take that conformer and try to calculate the descriptors. There are plenty of papers in ACS and very good papers in few other publishers, which is very well validated by the pioneers in QSAR. So please ensure that when you're using 3D descriptors, you have to be you have to be very careful. What is your starting geometry or the conformation? Most of the time, what I know is, as you have read in the slide itself, you take a 2D structure and you convert it to 3D either using OpenBabel, or even in Padel software you have an option to optimize the 2D structure to 3D structure using MMFF94 force field. Of course, they are something similar to universal force field, and the accuracy level is very low compared to many other model. So you might get a 3D conformation which might not be fitting into a biological conformer. So please ensure that whether you're using the right 3D structure when you're doing any 3D QSAR. Please keep that in mind. For 2D QSAR, when you're using 2D structure, no bias, no confusions there. You can start directly calculating the 2D descriptors. There are fingerprint descriptors available. There are many others. Anyway, coming to the generation of low energy conformers, that is where we have to be more careful. Ab initio is where DFT, Hartree-Fock methods, they are very accurate, much more accurate. Let me tell you, they are not the best accurate method, but the best accurate method we have more like CCSD, MP2, which goes a bit more beyond in quantum chemistry. I don't want you to do that in QSA because it takes more cost and more computing power. So, but they are best accurate. Your confirmations will be much best accurate in chemistry terms, not in biological terms. If you want it in biological terms, either you have to take molecular docking poses, or you have to take the pharmacophore conformers. Then comes with semi-empirical. In most cases, I have seen we are using semi-empirical. I have used mostly MOPAC. So MOPAC is again an open source tool which can be easily used it has a good accuracy for larger amount of parameterization, like if you have PM7 parameterization, it takes a little bit more time, but the standard one is AM1. In semi-empirical, we have issues with N triple bond N or some kind of salt forms and all. We have issues. So we need to clean the data set before doing any kind of semi-empirical calculation. Then comes a bit more low accurate, that is molecular mechanics, where we try to do with a low cost, which is MM1 or MMX methods, or FF94 force field based method, they are all molecular mechanics. They do optimize it much quickly, but your confirmations are not that accurate. The global minima won't be that much accurate. Now, going to the next slide, you can put your questions in Mentee already. Okay, so your Mentee is already there. In that page, there's an Ask a button. You can always submit your questions if you have any questions related to these slides. So coming to molecular descriptors, of course, as I told you, there are physico-chemical properties that describe few aspects of chemical structures. We also have empirical descriptors, which is like NMR chemical shifts, MP, log P, which can be also included as descriptors. For example, in many of my papers, I have included some manually calculated descriptors are there. So, which is very trivial because which I will be telling you in my next few slides, because relevant significance is very important. That means your descriptor choice should have some physical significance or some relation with your activity. If there is no physical significance and your statistical parameters are much favorable, that doesn't give any sense. So you need to have a balance of statistical parameters, as well as there should be a good physical significance or a physical meaning for your descriptors to relate with your activity. Very important. Now coming to the theoretical descriptors, what kind of descriptors? As I told you, 0D, that is constitutional, then 1D is hydrophobic parameters, and then 2D is topological descriptors. They are very important when we talk about some kind of what to say, migrational sites, or if you're I have few papers published on predicting the relative abundance of a chemical reaction, ligation properties, on peptidomimetics, where the cyclization could happen in a pre-organized conformer, even using transition state search. So topological descriptors can be used there, of course, very well, having good significance there. Then we have 3D descriptors. As I already told you, when you have 3D, the descriptors also change accordingly. We have geometrical descriptors, van der Waals descriptors, polar surface area, solvent accessible, and many other parameters, including quantum chemical descriptors like HOMO, LUMO, heat of formation, partial atomic charges, density parameters, and many more. And then when you go for a pharmacophore based or a molecular field based descriptors, then we have COMPHA, COMPSIA, which is being early used in many tools. Now they are out of patent, so anybody can use it now if you have access to it. So there are 3D field based analysis, including steric and electrostatics. Sorry for the typo, it is electrostatics. So steric hindrances, then I think you also are aware of van der Waals interactions and Coulomb interactions. Van der Waals interactions and Coulomb interactions also comes into hydrophobic parameters, steric and electrostatics, all comes into the parameters of field, molecular field topology based analysis. Now there are free tools available, which is having a data set should have 5 times of the descriptors used in the model. That is a rule of rule actually, but in most cases doesn't work out. So so don't stick to that. You need to always go for 5 times. Now CDK by Rajeshri Guha, it's a completely free tool, which is something similar to PADL. PADL is something widely used now. There are a lot of descriptors available. eDragon is an online platform, so that also can be used. Indigo RDKit is also available, but you can use it very nicely in KNIME, KNIME, KNIME software. So you have nodes available for Indigo RDKit. And there are plenty of other commercial tools also, which I do not want to address it here. Now dataset and classification is also important. Let's say you have n data points of biological activity. Now we need to do a little bit of filtration, tautomers, then like salt forms, small fragments and big fragments in a single compound, all these things to be filtered actually. Now here I did docking, so docking analysis. So I just and also I was looking for oral bioavailability. So I was looking for Lipinski rule of 5, natural charges, as well as filter based on spatial target protein. So I did a docking analysis, which was the best poses. From there I picked up my 3D structure. and then I removed few compounds based on the filtering. Then I tried splitting my models into training set, test set, and two different external set. This two different external set you can even split into one also, that is fine, but three data sets, very important. Training set is mostly used for us to build a QSAR model, and it predicts R-square. The test set is something which is also called as prediction set. We try to predict its values using the QSAR model based on the training set to predict the Q square. The external set, which tries to again use the training set models to again, you can say, external R square. So why we are trying to split it into three? We wanted to revalidate both internally and externally so that our models are reproducible and reliable for the same set of chemical class. Please be very careful when I told you same set of chemical class. QSAR models are done in two different set of structures. One can be homologous or local models. The second can be diverse models or very global model. That means your nuclei or the main scaffold of your compounds all will be same. There will be small derivatives on the sides. They are called local or homologous. Diverse set is the scaffold, the nuclei of the structure is completely different. They're completely diverse. When you try to do a clustering, there will be a lot of singletons which they don't match each other. But on the homologous, they all come under one single node because they are all clustered and they are similar. So we should be very careful on choosing the right descriptors for and also right method for different types of models, like whether your system or the structures are homologous or heterogeneous, that is diverse set. That is very important. Number of compounds chosen for these two will be also different. For local models or homologous models or structures, the number of structures could be minimal. Minimal means I personally prefer at least 25. Personally prefer. I have seen some models with 8, some with 10, some with 12, but I personally prefer at least 20 to 25 for a better reproducible data. So keep in mind QSAR is completely used to help others to design new molecules with our models. If your models have more inconsistency, if your model has more inconsistency and it is not reproducible, then what is the point of generating these QSAR models? So you should be very sure that when you're using for homologous models, ensure that you have a good like applicability domain, and you have at least 20 to 25 compounds with the biological or experimental data to generate these models available. The next part is heterogeneous or diverse or global models. For that, minimum you need 60. That's a best thing, at least 40. So 60 is recommended, at least 40 is also required. Otherwise, your diverse, the coverage of chemical space won't be covered. Now, in earlier papers in 90s, most of them have preferred only local models, homogeneous models. But when we have sophisticated models, softwares, on a click of a button, we're getting all the workflows being done. We started playing around with the diverse set. But diverse set is always recommended for better coverage of diverse class of chemical molecules. That is always recommended. But you have to take care of a lot of statistical parameters so that you don't have false positives, because there are more chances of false positives in diverse sets. So you have to be really very careful. Okay. I recommend everybody to be on mute if someone has recently joined. Okay. Anyway, so going to the next slide, what are the different methods that we have for feature selection? As I told you, feature selection is something that we want to choose which is the right descriptor that to be will be better correlate and relate my activity. That is the thing that we are looking at feature selection. So we are looking for MLR. I don't recommend MLR for diverse set. For the homogeneous models, they are much okay, because the problem with MLR is overfitting is there. Too much of overfitting issues are there for linear regression methods. So it's better to use PLS for models with overfitting and all. So there are many things to be considered rather than R-square or regression models when you're trying to look at which model is more preferable and recommended. Right. So best MLR is that it is a combination of genetic algorithm with MLR is called best MLR. So it uses genetic algorithm for feature selection, and that descriptors will be used to correlate with the activity using linear regression. Then we have PCA, that is principal component analysis. Here what we do is a collection of descriptors are being converted to factors, and multiple factors forms the models. So here it is superior than MLR in case of number of descriptors in the model. So you can have more pool of descriptors into the model, where, as I told you, more number of descriptors will be considered as factors, and they try to correlate with the descriptors to the number of compounds in a model. Then coming to PLS, that is partial least squares, we have descriptors are extracted into new component as to maximize the correlation, and this model has multiple dependent other parameters. So these are the different statistical methods that is available. Then we have also many more machine learning models available. So let me, I have not put that into the slide. We have machine learning models available is like, which is like random forest. Then we have Gaussian process regression methods, then SVM methods, neural network methods, but again we have to apply it on different kinds of cases. For example, Gaussian process regression method and random forest can be applied in all these data what I was discussing about. And Gaussian process regression method, I don't know how many of you are aware of. So these are available in WEKA, WEKA. WEKA is a program which will be also used in machine learning and data mining analysis. Now coming to model validation, as I told you, be very careful in selection of descriptors. Don't think that whatever software has given descriptors that would be right, because you have to iterate this process. Model generation to be iterated several times, and the feature selection should be iterated several times. They come up with plenty of options just based on R-square. They say this is the highest R-square, these are the number of descriptors which is dependent. You might think that is the best one. No, there might be below other descriptor collection which is having very less or maybe bit lesser than the highest R-square also. That doesn't mean that the model is bad. So ultimately you should have a careful selection of descriptors, because you need to choose and understand based on the physical significance. So ensure that your R-square is also balanced. If your R-square is 79, nobody is saying your model is bad. Or if your R-square is 84, 86, 72, it never said to be this bad. If it is 99, 98, yeah, something you have done to make it 98 or 99, so that is bad. Or if your model is like 50 or 52, 54 or less than something 60, of course not acceptable. So that's where we are trying to understand. So you need to have a better reliability and reproducibility on the data. And the significance of the descriptors with the activity is also very trivial. Optimum number of compounds is mandatory, as I already discussed last time. So here we are talking about the fitting, that is the R-square, and then we come up with a QSAR model where we are doing cross-validation on the subset, iterative cross-validation splitting, and cross-validation on the test set. So that is why I said there are two different methods of internal validation, that is Y-scrambling and also randomization. So we try to do several iterations of validation. Leave one, leave out, leave many, and one out. So that is what LMO and LOO. So we try to do different iterations of LOO. For example, we leave the first one, take the second, leave the third one, take the fourth. Like that we start with a different set. We leave the second one, take the first one, leave the fourth one, take the third one. So like that we try to iteratively come up with a model for internal validation. Most of the time when you are using commercial tool, they do it automatically for you, but I recommend you have to understand this process. Otherwise you cannot write the discussion part of your QSAR. The discussion part is a take-home message for QSAR papers. It's not the R-square values and other things. What is the significance? What is that I am going to get from this model if I use this model to predict for my new structures. And then you have a prediction set or an external set where we do a external evaluation. We understand applicability domain. Applicability domain is a very important factor. Sorry, I'm not sure whether I have a slide here. Applicability domain, what we try to say is, let me see whether, oh, I cannot scribble here. Okay, sorry about that. So what we are trying to do is, let's say you have consider a ball or a circle. Inside that circle you have training set, and yeah, you have your training or your QSAR model structures there with the value points. Now you brought in your structure, you predicted the value. If your structures is fitting within that ball or circle, then we say it is within the applicability domain, or your test set or your prediction set or your external set fits within the training set circle, then we say your models and your predictions are within the applicability domain, and then we claim that yeah, your predictions are reproducible. If your external set is outside that circle, then it is not within the applicability domain. That means your models are not reusable. So that doesn't mean that even though if you have an R-square of 84, if your model is not reusable, there is no point of using or publishing that data. How this can be analyzed? How applicability domain can be analyzed? So there is an Excel sheet. Of course, I have it. I can share after this if someone is requesting for the same. So we have to calculate the hat leverage. The hat leverage value tells you the barrier where we have to set this particular domain, the applicability domain. And we will be plotting a graph which is called as Williams plot. So Williams plot is used to visualize whether your training set applicability domain actually has your external as well as prediction set within that domain. If it is within that domain, your models are good, they are reproducible, and then only your QSAR is complete. Now most of the time I have seen many papers, we have seen only training set and test set. So you predict cross validation fitting as well as the training set fitting, that is R-square or Q-square, finish it off. That there we are doing only internal validation. We have to do external validation, and in both the case applicability domain has to be carried out, and then only we have a justification these models are reproducible. Okay. Now this is something that I have done for one of my work where we have used different tools with the combination. For the data preparation, of course, we have did a Tanimoto similarity analysis to understand overlap of data and others, and the k-means clustering was done. And for conformational search with target, of course, we have used OPLS as well as MMX, which is available in PC model. PC model was earlier commercial tool, but since 2019 December, the author said he is getting aged, so he wanted to give it out for free. So PC model is a software where you can integrate MOPAC, MMX force field, conformational analysis can be done using GMMX parameterization, and there are a few other integrations available. It's a small piece of software. Now it is free. So while I am doing it, I was using that for my conformational search. Of course, docking also was done to match it. And then for where I couldn't do docking, I took with the AM1, which is semi-empirical, where geometry optimization, charge distributions were being done for parameterization. And then I did the calculation of descriptors from different tools, and then I tried to merge them. So merging has to be done very carefully. There shouldn't be duplications. There should be independent descriptors which doesn't correlate among themselves. That is very, very important. Intercorrelation among the descriptors can also lead to false positives for QSAR modeling. So ensure that there is no intercorrelation between the descriptors, maybe somewhere around 85 to 90%. So if it is more than that, you should avoid those descriptors. Now, which one to avoid, you have to choose. That is very important. So most of the time we just click the button, get all the descriptors, let the software decide it. No, we have to actually analyze what descriptors are having intercorrelations and all. So I don't know how far you have mostly done, so it depends. And then we have to do a descriptive statistics, which is the feature selection, where we do the trimming of descriptors. Then we try to apply different methods. As I told you, combination of genetic algorithm and MLR will try to give you a correlation. If you ask me, here I have done more than 680 iterations for my modeling to come up with the final one. That much iterations I have done on all the model, because different combinations of training set and test set to see which one, because ultimately we have to validate it internally and externally, doing scrambling, scrambling method and other methods. So everything should fit in properly. It's not only the R square, my Q squares and external validation, everything should come properly in the model, otherwise it's not good. When a single compound is changed from your training set, your coefficient of the descriptor will change. When your coefficient change, your activity or predicted activity will change. So that's where I said we do several iterations, and it has to be done. And then we also check with applicability domain, and then reverse QSAR. There are merits and demerits also. Significance or role of chemical composition on activity, where we try to correlate them with the significance of descriptors. And models can be used to predict activity for novel descriptors, novel compounds. That's where I told you the use of QSAR. And then coming to false correlation, of course there will be experimental errors also. So keep in mind, might be you have done everything, but there might be false positives. That may be because of experimental errors also. We don't know, really don't know. Sometimes a statistical point of view, you will be able to find what were the experimental errors also. There are papers reported when they did QSA, they found that there are some experimental errors. And the main thing, GIGO, garbage in, garbage out. If your structures are not good, if your normalization of your activity is not good, if you are trying to combine different paper data, I mean different lab data together to make a single QSAR, and you did not take precautions to understand whether their bioassay are same, whether their targeted targeted proteins are same, whether the assays have similarity, and if the units can be converted to make unique, same units. If you did not take care of all those things, then you will have issues in your model. And in some cases we would have seen the IC50 values put as greater than 1500 or 2000. In that case, what we do, we just leave that greater than and we put 2000. How can you know that it is 2001 or 3000 or 4000? We don't know. It's better to avoid those data. Usually it is better to avoid that data. So these are the things that might prone to false positives, and the models are not reproducible. So you have to be very careful on garbage in and garbage out, and there are many more. I'll refer to a paper which was published I think 2014 or 16 by a group of people like Professor Grammatica, then Tropsha, then John Dearden, and few other people. So they have written a review in ACS. It's about 32 pages where they have clearly mentioned about all these parameters what I'm telling you today. Now, not better or worse, it's a complementing. Two descriptors which are orthogonal can give a good view on different things. That's what I already told you about intercorrelations and different descriptors. The significance will be different. They might be having same values and they are orthogonal, but it has a different perspective and views on the models. So you should be very careful when you're trying to do QSAR. So that's all from me for the presentation today. Now you can start questions. Now I will be doing a demo, a live training session, but not today, maybe a bit later once you read and understand about QS, because that is very important. Everyone will be urging to do the software, but most of the time we don't really look into the theory to understand what to do and what not to do. That is the title of today's session, do's and don'ts in QS, because you have to understand first before touching a software to do QSAR. Okay, so here are the questions. So once we go through the questions in the mentee, if you want to submit again, you can always submit. As I told you, you can go to this particular website, mentee.com, and you can type key in the code 741474, and then you just refresh the page. You will be able to, at the bottom of the page there will be ask a question, and there you can ask the questions and it will come up automatically. So let me now go through the questions now. How easy is QSAR? Let me tell you, QSAR is not at all easy. One of the most difficult and very cautious method that you have to use. On each step, human intervention is required. On each step, your expertise to be put in to make decisions and selections. Whether the structure got the right conformer, whether my starting geometry is the right one, whether the descriptor calculators are correct or not, whether same descriptor on different softwares giving different values or not, which one I should take, whether the models generated on different iterations giving a different R-square, now which one I have to take. So it's not at all an easy process. If someone is saying easy, sorry, that you have to clarify with them. For me, no, it is a several step process and you have to validate each one. If you have done that, then a QSAR will be liked by organic chemist and medicinal chemist. If you ask me now, most of the medicinal chemists and QSAR chemists or organic chemists who really know things which from their past experience, they really hate QSAR, unfortunately. The reason is we did not put it in front of them in the right way. So if you can come up with a very good reproducible model, that's where a chemist will get convinced, okay, this model is helping me, whatever structures I'm drawing and which is in my mind, it is able to give the proper data because I tested it in biological bioassay and it is giving as my expectation. Don't think there will be 100% matching or fitting. No, at least something it should make the trend. That helps them to make use of your QSAR. I hope I answered that question. Now next one is, which one is better, commercial tools or open source tool, and what is the difference? See, for me, my personal view, nothing on the other side. For me, both softwares are good, both softwares are bad. I hope you got it. Both softwares are good, both softwares are bad. It is depending upon the person who is using it. The only difference with commercial and open source is commercial. Today, if you write a question to the support team, you get an immediate answer within 24 or 48 hours. There's a person to answer your custom questions. On open source, you have to put it in a forum. You have to ask somebody with your friends or in the community. There's nobody sitting there to reply to your questions because it's free. Nobody is being paid. So here, commercial tools, you paid for it, so you get support, a tremendous amount of support on how to carry out your analysis. So that is the main difference. The other difference on coming to accuracy, I don't completely agree with commercial tools will give better accuracy and open source doesn't give accuracy. No, nothing like that. Both give accuracy and both gives very wrong data also. As I told you, it completely depends upon how you intervene on each step and checkpoints and you come up with better next steps decisions. I hope I answered that question. 2D or 3D descriptors, which one is preferred? My personal preference is always 2D descriptors because there is no bias. Of course, when you include 3D descriptors, you have plenty of accessibility to more descriptors, but bias is also increasing. Whether the geometry or the conformer that I have taken is a right conformer or not. The geometry that I have taken is actually a biological conformer or not, either through a pharmacophore analysis or through a docking analysis. Or if you're just generating conformers just based on some ab initio method or semi-empirical, it is a chemical descriptor. So why I am telling you this, keep this in mind, very, very important point. When you have a drug in your hand, or you synthesized a drug molecule, API, the active pharmaceutical ingredient, that particular compound is having a conformer, a crystal form. But when it goes to the biological system and binds to a protein or any target, the conformation changes. That is called a biological conformer or a biological complex conformer. We do not want the synthesized molecule conformer to do the QSAR. What is the point of doing that? Because we are doing the biological activity data, right? We want a biological complex conformer. That is very important. If you are able to get that biological conformer and then calculate 3D descriptor, there is some sense there. Of course, there will be errors, fine, but at least you took a right direction to calculate the 3D descriptors. If you are not able to do, it is biased. So 2D is more preferred because you don't have any confusions there. But yeah, limited descriptors, not limited, there are plenty, but still you have to find the physical meaning of these descriptors. That is a problem, not a problem I would say. Some people will find it, of course I have also find difficult in some of the descriptors in Padel as well as in eDragon. There are 3000 descriptors and 1800 descriptors. Some descriptors I am not able to find the physical meaning. Unless I find a physical meaning, how will I write my discussion saying that what is the significance of this descriptor in my activity? If you will see, there will be plus, minus, 2 part, 3 part of a same descriptor. What is the significance? So you need to have an understanding about the physical meaning of the descriptor. So your choice of descriptor, ensure that whatever software you are using, your descriptors have a physical meaning and significance to correlate to the activity. Otherwise, you will find a big problem in how to write your discussion part to convince the reader, or when you're going for patenting or anything consultancy work, you will be finding it difficult how to convince the experimentalist what is the significance of descriptor with this descriptor with your activity. So the next question, which tools are used for external validation? It's basically not tools. I have used KNIME, KNIME. It's a pipeline based workflows, and then I have also used QSARINS, which is from Polar Grammatica's group. I have extensively used and published papers. It is free for academics, but she only gives to people who has a bit experience on QSAR and who have published good papers. So when you're requesting for license, you have to cite your QSAR papers, and then only she will give you the license for free. So it's a software which is included. Padel is also included into it, and it also follows whatever steps I have mentioned here today. Please share the resource and details via email for a novice person to start with QSAR. So let me tell you, what you can do is you can send an email to me I will put it in the chat. So you can send an email to this particular email address so that I can respond to you. There are few emails to be responded from the previous session. I'm still waiting for the resources. I'll be sending it to you. So in the chat you have the email address, so you can send an email to this so that I will respond to you with the papers and other details. Next question. Oh, cannot see a PPT. I think that is being solved. Earlier you have mentioned it. What pipeline you follow as a professional with reference to tool or script from feature selection to model? Okay. I told you. I think I answered during my slides. From feature selection, because when you're coming to feature selection, the genetic algorithm doesn't give you only one model. It will give you from, let's say, 2 descriptors, what is your R-square, 3 descriptor, what is your R-square, 4 descriptor, how is R-square. So we give an upper limit, what is the maximum descriptors that particular model can. Lower the descriptors, it is the best for any QSAR model. Don't try to put in more descriptors there. Again, depending upon which method you are using. For MLR, lowers the descriptor, better the reliability of the model. But if you are going for PLS or PCA or any machine learning methods, you need to have a good significant descriptor, number of descriptors in the pool to do feature selection. That's where you have to keep very careful thing. There's no script, unfortunately. The software just predicts the descriptors. Let's say 1800 descriptors. Now you have to sit and choose which one I have to choose. Don't choose everything. Unfortunately, no, I don't recommend that. Please have an understanding what is the descriptors in your pool. Now you will say, oh, it is difficult. Of course, yes. If you want your QSAR to be cited, models to be cited by hundreds of people, and it is to be reliable, you have to do this task, no other choice. There are tools and scripts that can be done, but again, that is more of a statistical parameter. But I clearly told you, physical meaning and physical significance is very important when you are applying it on a biological or bioassay data. If it is a chemical or a physical data, a little bit of leniency will be there. So feature selection to model, you have to try with 2 descriptors, 3 descriptors, and 4 or 5 or 6 descriptors, whatever extent you are going to go, and then try to model with all possible iterations or different models the feature selection has been suggested. You try to model most of them, and then from there you choose the best ones. That is the point. Now feature selection also has a glitch here. Now when you're trying to divide, as I told you, we have to split your training set into training, test, as well as validation. In some software they allow only training and prediction or test set. When you're doing the splitting, on what basis you're splitting it, that is also very important. There are certain methods that to split. One is randomly, second is based on cluster, based on clustering method, or again Y randomization, leave one, leave out, different methods. Now all are feasible. Now you have to try all. That's a case I told you, you have to do several iterations to come up with a single model. As I told you, for one of my work, I did around more than 680 iterations to come up with the final model. Unfortunately, that is the only go. It's not a single iteration job to get the best QSAR model. Power MV. Power MV is a pipeline, of course, available with MGL tools, but I have never done any scripting on Power MV. As I told you, I use more of KNIME, which is where many plugins and nodes are already integrated. For example, Padal descriptor nodes are there. Indigo is already there. RDKit is there. CDK is there. Weka is there. Many statistical parameters from R programming language, it's already there. Machine learning models are already there. So I am more using KNIME than Power MV. Sorry, I'm not sure with Power. I'm not used in MGL tools. I mostly used ADT, that is Autodoc tools, then Autodoc Vina, Autodoc, and a little bit of their CAD pipelines. Could we have a small project for better understanding of the subject? Of course, yes, but as I told you, before going through the project, you have to read plenty of reviews. I don't want you to read any research papers. I don't want you to read a research paper before understanding what is the real QSAR. As I told you, please send me an email, which I have already shared in the chat. Please send the email to me so that I will respond back to you with references to the reviews. Please read those reviews. It takes time. Understand the subject concept first, and then you can start, because most of the time we get stuck when we start a project and it is not going anywhere. We are not getting the right models, we are not getting the data, we are stuck in between, and then we start reading what could be the possible solutions. Why can't we have this reading earlier, expecting all hiccups in front of us, having plan A, plan B, plan C, or plan D? In QSAR it's like that. I have ignored around 18 projects because I don't have data to come up with the QSAR project. So if some data comes up, those projects will reopen and it will go ahead. I don't have data. So that is how it is. So better to have a better understanding of the concept of the subject, and then decide on which data you have to work. Then you search for data. If the data is available, then only think of QSAR. If you don't have data, don't think about QSAR. So most of the time, okay, I will do a QSAR project. Fine. On what you are going to do? I'm going to do in cancer. Fine. Do you have data? If you have yes, good. If you don't have data, then why do you think of QSAR? That's a question. So better understand the concept and required data. If the data is available, then think of QSAR. Otherwise, sorry, no. You should be very careful in doing QSAR or choosing the data. What are the major difference between QSAR and pharmacophore modeling? Yeah. So see, QSAR, pharmacophore modeling, similarity search analysis, clustering, these are all ligand based approaches. When you really don't have a protein structure data to do a structure based design like docking or many other things, if you are not able to do it, that's where you do QSAR or pharmacophore modeling. QSAR, your data can be anything. From a structure, what all you can extract, you can extract. Even pharmacophore features also can be extracted in hydrophobic terms, in van der Waals, many other parameters. And you are quantitatively trying to, numerically you are trying to correlate your structure, numerical data with your biological or bioassay or biomedical or whatever data, and that's where you quantitatively compare the relationship and try to understand. That's where the QSAR comes. When you're going for pharmacophore modeling, ensure that you have a reference molecule with you. Let's say you're working on any specific, let's say malaria, and you pick up a reference molecule which is already in the market, a FDA approved drug, and you picked up that compound, and you don't have activity for that data available, you don't have activity for your structures also available. Let's imagine in that way. Now you wanted to screen all your database compound, let's say 1000 molecules. You wanted to see whether the pharmacophore features of your reference molecule, that is a FDA approved drug, it could be negatively interaction points, positive interaction points, hydrophobic interaction points, lipophilic interaction points, phenyl centers, and many more. Excuse me. There are many pharmacophore mapping available or features available. Based on that, they will try to first generate the pharmacophore map of your reference molecule, keep that as a reference. Then what they do is, from your 1000 molecules, each molecule, they try to generate conformations. Of course, on a chemistry point of view, they generate conformation. It can be 1000, it can be 500 conformations, we don't know. Based on the algorithms, there are free tools for conformation analysis. One is, I already told you, PC model. The second one is Balloon. Balloon is a command line script but does the job. And OpenBabel also has an option to generate conformation. So OpenBabel, Balloon, and PC model are the free tools for generating conformations on molecular mechanics level. So accuracy will be not that great, but at least you will get the conformations. So after generating the conformation, now you try to generate pharmacophore for each of the conformations. Now the pharmacophore of your molecule, I said 1000 molecules will generate, let's say each one 5 conformations, you will have 5000 conformation molecules. Each of the molecules conformers pharmacophore also will be generated. It is called pharmacophore map or interaction points. That map will be made a similarity analysis with the map of your reference molecule. So all that 5000, they try to come and go and try to see which conformers pharmacophore features are close or matching with the FDA drugs pharmacophore features or map. That is called pharmacophore screening, actually, not modeling, pharmacophore based screening. There we are trying to find out what are the compounds in our database of 1000 molecules which can have similar pharmacophore features of the FDA approved drug. This we do when we don't have a protein, when we don't have any biological activity data available. So these are the major differences between QSAR and pharmacophore modeling. I'm not going much deeper, but I think this is the best explanation that I can give. The next question, is there a possibility to do a virtual project on QSAR to learn it and work on it? Absolutely yes, but if you wanted to work under me for a project, you have to give me presentation on 5 different topics on QSAR. Once the presentation is done, yes, of course, I'm happy to do give you projects. There are about 68 students have done project with me. A few have done on QSAR, a few have done on pharmacophore, most of them done on molecular docking in combination with QSAR. Everybody knows that they have to do a presentation because understanding the subject is very important before starting a project. So please understand that subject. Whoever is interested, please write to me or you can WhatsApp me, and then I can give you the topics and you can give a presentation. Of course, you can give a session like this, no problem at all. You can have a session with me. If everything is good, you can give a session like this, so others also will learn. It's all it's a sharing. Knowledge sharing is the best deed that we can do it right now. The next question is, as I'm a beginner, how do you suggest to go about QSAR to be good in it? As I told you, you have to read a lot of reviews, the good reviews, very good reviews, very important. Where you start is very important, okay. And take away the assumption that QSAR is easy, just to click of a button. No. So Please read reviews first. It will be boring. It was too boring for me also. But unfortunately, we have to do something, right? We have to apply it somewhere. We need to actually know its merits, demerits, or you call it as advantages, disadvantages, or do's and don'ts, whatever it is. You have to know before touching it. That is the best part. Now you think, okay, during the project I will learn it. Sorry, during the project when you learn, it might be too late because you don't have that particular data available. You have to trash it, and you have to again from scratch you have to start your project. It's better that as a beginner I would recommend please read reviews, and also try to attend few online videos already available in YouTube. Plenty of them are available. There are very good ones. Please try to listen to them, and once you are sure, try to prepare a presentation by yourself, at least for 20 slides. Make sure that you also understood that, and you are able to really understand and make a decision on what things are right and what things are wrong. How easy can we do SAR studies in QSAR? SAR studies in QSAR can be done when you have a better understanding of applicability domain. So when you have a better understanding of where your prediction fits within the training set, that's where you can do a better SAR studies. Of course, it is possible. Thank you so much for a very nice session. Just a clarification. Do we need to consider some inactive compounds during validation to check if the model is able to differentiate? Oh, this is a very, very good question. I really missed to mention that. Of course, yes. So your training set, when I mentioned about homogeneous or heterogeneous set of structures, I did not mention that you have to choose based upon your activity. I have seen some papers, they try to arrange based on activity. The most active ones they picked into the training set, and non-active ones into the test set. It's really wrong, not the right way. You need to have a diverse set. You need to have both inactive, moderately active, and good active compounds. It's very, very important, because the best part is on initial guess, we need to be very sure that you have a diverse set in your diverse class of compounds in your set. It's very, very important. Then only the reliability and reproducibility of your QSAR model is viable. So diversity is very important. So keep sure that you have both inactive and active molecules, and the model need not to differentiate it properly, because your applicability domain will show you which one is inactive and which one is active. Of course, it will differentiate in the applicability domain. So if your predicted molecule is very close to inactive molecules bunch, then we say, yeah, your molecule is inactive, the newly designed molecule. So applicability domain tells you a bit more than your scatter plot. What is your view on auto QSAR provided by many big companies like Schrodinger? As you said, in each step we have to whoever software. Schrodinger is a big company? I really don't know. There are many other companies. The problem is that we who have to choose it. Auto QSAR is there, Auto Modeler is there, QSAR is there, then Farm Screen is there. Plenty of softwares. Now, the question is, which one is reproducible? So, whoever asked this question, I am sure that you have access to this particular tool. Take a paper, a published paper, try to rebuild the model, what they have already published, okay, rebuild the model, and then try to bring similar structure from a different paper where they have done only experimental data, try to predict the activity of that structures which is only having biological activity data, no QSAR being done, and you predict it. If the R-square for the predicted one with the biological activity one is greater than 64 or 65 R-square, I'm talking about, kind of okay. That is what is acceptable on statistical terms. More the better. But if it is lesser than that, it's not reproducible. So it's not about what software you used. You have to test it manually whether it is reproducible or not. So that's what we do most of the time. So when we get a project, if someone is saying that this is done, this is good, to evaluate that software, we try to reproduce the data, whatever they have published. If we reproduce and we are getting the same data and for a experimental only data, and we try to predict using this model, whatever they are published, we using that model, if we are able to get somewhere closer to that experimental data, then that is a good model. So it's not about which software you have used. So, and any big company or any big software you're using, each step have to be monitored. I know in these tools we have workflows available. You just click the button and go. But there is a drawback there. Now tomorrow, let's say if it is a PhD student who is using this, and on your Viva voice, someone is asking in between step. Are you going to say that I clicked this button and I got the result? No, that's not a great answer, right? So you need to have a understanding of each step what the software has done. See, software is made user friendly for people who don't have time. Basically the people in the commercial sector, they don't have time, they want something to be going very fast and easy. But as an academic and researcher, we need to have time to understand the science behind it, right? So no black boxing is entertained, especially in QSAR. Is Padel a good descriptor tool for prediction? See, I told you, there are plenty of descriptors that can be calculated. Manually also descriptors can be calculated. It is about the feature selection that you are going to do. Padel has fingerprint descriptors, 2D descriptors, 1D descriptors and 3D descriptors. It is about what is it, which descriptor is significant for your model. That's where I wanted to tell you that putting in more descriptors and making the software to build a model is not a good thing actually, because software just looks at numbers, nothing on physical significance. So you need to have an understand what type of descriptors I have to choose, because for example, one of my student, PhD student who is working on aging, the paper is already online now in Life Sciences Journal. So there, what we have done is we used around 18 different descriptor tool generation tools, and all we did feature selection. From the feature selection, we created a pool, and then we try to do models. And most of the descriptors were not good at all. And the reason is we couldn't find any relationship with our activity. We were looking for anti-melanogenic activity. So we couldn't get any significance of the descriptor to relate to that particular activity. So that activity has specific need of the drug molecule should have. So we wanted to relate that descriptor with that activity. So that's where we have to decide. Padel is a good descriptor tool, no problem for a beginner. But as I told you, as you go on, you have to make your better decisions. Descriptors are based on biological activity. How to find this biological activity data? Oh, come on. This is which I missed to tell you. See, biological data you can either find from Chembl, which is a very well curated data. You can even get it from binding database to some extent, not everything. Then, as we have done in 2011 and 2010, you have to manually go to paper, research papers, draw the structure, check their table, make an Excel sheet. That's what we have done earlier. Nowadays you have a lot of curated database, you can do that. Descriptors are not based on biological activity. Descriptors are based on the structure that you have taken. What I am trying to say is when you are generating a model, QSAR model, that particular descriptors which you have chosen for modeling should have physical significance with the biological activity. So as I told you, there are several models will become, so there should be some physical relationship or physical meaning to correlate your descriptor with the biological activity. Sir, can we do QSAR studies without doing? Of course, yes, you can do without doing. See, I mentioned docking studies and pharmacophore based or ComSAR, ComSIA methods only to make sure your 3D structure is having a good geometry for you to calculate the descriptors without any bias. You can always do QSAR on 2D, and now you can always do QSAR on 3D if it is more of a chemistry terms or physical terms. Means if you are looking for some chemical properties or physical properties, not physicochemical properties or biochemical properties or biological properties. Yeah, it is I cannot generalize it in that way, but that is the best practice that you can try. So without docking also you can do QSAR studies. Which software is best for QSAR model? As I told you, the QSAR model, one single piece of software cannot do. Even whatever commercial softwares you are telling, even though one name is given, but inside several processes is being going on. You have to convert your smiles or structures to a proper structure. From there, you have to calculate descriptor. So the first conversion, generating, all will be done by one tool. That can be with OpenBabel or RDKit. Then you have to calculate descriptor. The descriptors can be calculated using Parallel, CDK, Indigo, RDKit, then many more are there, and a few commercial tools are also there. But as I told you, here we are not doing any promotion of any commercial tools. Mostly we will discuss about free tools. Commercial tools are also there. Then after the descriptor calculation, now we have to do pruning, like not pruning, like trimming of the descriptors, like feature selection. For that you can use genetic algorithm. Now genetic algorithm is available in 9, KNIME 9. Then Orange is also a data mining tool where you can use machine learning and statistical methods can be used there. Then you can use Weka, WEKA. These three tools can be used for generating models, feature selection, finding out intercorrelation methods. All these can be used by. So I just set a pool of softwares on each different stages. Now you think, oh my god, I have to learn all this. Yes, unfortunately, yes. If you want to really know the science of QSA, you have to know about this. How can we study physical significance of descriptors? Very good question. So each descriptor, for example, if you go to Padel, in their website, they have given an Excel sheet which tells you the references for each of the descriptors. I don't know how many companies have given that. Okay, all these big companies or commercial companies, if they don't give, you have to ask for that. If the descriptors significance is not given, that means a reference is not given, how do you know the significance? That is the sad part. So that's where it's a very good question. So when you have a descriptor, there should be corresponding literature or publications. Of course, Padel is giving that. I really know that. They have an Excel sheet which gives you the references for most of them, not all of them. And others, Google it or put it in Google Scholars or in PubMed or in any literature search databases and try to find out, go to the older literatures to understand whether someone has discussed about this descriptor's physical significance. It's very, very important because that is the bread and butter for your discussion part of your paper. Otherwise, what you write in the discussion on your QSAR, you will just write about R-squares and other things, right? But more we have to focus on physical significance. What are the criteria for selecting descriptors? The criteria, as I told you, for example, let me give an example what I have worked on. I have worked with the US Department of Agriculture for my PhD. So we published a paper as promising mosquito repellents for Aedes aegypti, that is yellow fever mosquito. So we took in some of the US Department of Agriculture's library data and few other carbamicides. These are all published, it's in PLOS ONE and also patented on attractants, the group. Then we try to analyze minimum effective dosage, MED values, of course experimental data, and also people volunteered to put your hand with a sleeve opened with some repellents inside a box full of mosquitoes to understand how many of them are sitting and staying away, how many of them are dying. It's just understanding the numbers. So from there, what we try to understand is, what is the property that we require for a repellent? It is maximum coverage in the environment. In a room temperature, how far this repellent can travel on the air, on the atmosphere, on a particular pressure and a temperature, so that the mosquito doesn't come to that particular area, as we do for all the other mosquito repellents, whatever we are using. So what is the property that we are looking there? That's what we have to understand. And try to correlate that property with whatever descriptors that is being calculated. It could be quantum chemical descriptor. We picked up a quantum chemical descriptor to correlate how far that molecule can travel within this particular temperature and pressure to maximum coverage. What could be the property dependent there? So that is the way of understanding you need to have. Even on biological property also. For example, if it is a cancer or let's say something else, it has been prone to zinc metal ions or something for copper, or hydrophobicity should be more for this drug which is going to bind or inhibit that particular a function of the protein target. So that's where your level of understanding. Could you post certain reviews for no? Of course, please email to me, which email address that I have given you in the chat box. I will refer you to very good reviews for you to start. Please given free soft. I already told you the free softwares. Hope you have noted it down. This is also YouTube recorded, so you can always check YouTube the link that I have searched if you forgot it. So you can look at it. Then can you suggest few topics on just now told so that I can make a PPT and send you? I mean, oh, that's great. You already made a decision that you have to go with it. That's great. So the topics, as I told you, I recommend you to email. I will email you. There are few things to be discussed with you because of a level of understanding. Expectations, because my expectations I am looking not for black boxing on each part that to be done. So the topics basically will be understanding of descriptor calculations, then understanding of methods used in modeling that can be bifurcated as statistical methods and machine learning methods. In statistical method, for what kind of data we have to use which method. For example, PLS. For what kind of data I can use PLS? For what kind of data I can use MLR, multi-linear regression method? For what kind of data I can use neural network? For what kind of data I can use GPR, Gaussian process regression? So you should be the decision maker. Based on this data available, I'm going to choose this method because the other method is not right for this kind of data. So these are the kinds of topics that we have to make a better understanding, so that when you know the data, you are very focused. Okay, this is the data, this is the only data that is available. I am very now sure these are the methods in front of me to try it out. That's a good question actually. Then what is the next? We have one more question to go. Tool to calculate protein descriptor. Okay. I recently saw in GitHub where they can calculate protein descriptor. Now it is time-taking process, but what I understood from the time I'm doing QSAR, I've always thought of why can't we calculate protein descriptors. But that time I've read some papers using molecular dynamics minimization minimization. You will be able to extract few properties from that for that particular protein. One. Second, on the protein sequence, there are several bioinformatics tools available where you can extract few data, numerical data, categorical data of to be considered as protein descriptors. Third, when you are trying to focus mainly on the binding side, where ligand you are trying to dock, you can do QMMM or catalytic site studies, and from there you get a lot of numerical data can be included as a descriptor. So any numerical data that you wanted to do quantitatively, that can be included as a descriptor. So I'll try to share. I don't know who has asked me. Please email me. I have posted my email address in the chat box, so that yesterday when I saw this, they have come up with a tool, a script actually, not a straightforward GUI tool, who can calculate the protein descriptors. Okay. Great. So I have already answered all the questions, but if anybody wants to ask any questions through audio, you can unmute yourself and you can ask me. Otherwise, we will end up the session soon. If you have any questions, you can unmute yourself and ask me if you have anything. I think someone want to ask any question? No? Okay. I think none of them have any questions. Now we have two more sessions coming this week. One is on Tuesday, April 4th. You please go back to Mentee and you can give, are you going to attend these both or one? It's up to you. So you can choose the one. So coming April, Tuesday, 12 p.m. we will be having a systems more of a preparative on COVID. So COVID on a genomics perspective as well as on a proteomics perspective. That is more of a bioinformatics perspective. We are looking at COVID, and The resource person will be from Birla Institute of Science and Research, Jaipur. So I'll be sending you the details later. And then on Saturday, April 18th at 5 p.m., we have another session on systems pharmacology, that is polypharmacology. For example, if you're working on natural products, or if you're working on plant extracts and you wanted to identify the possible targets, and also wanted to do target identification on understanding what are the possible relationship and interactions between two different genes or targets, protein targets, that you will be able to find out based on systems biology. So that we will be focusing, and a faculty expert from Vellore Institute of Technology, Vellore, will be joining us on Saturday, April 18th at 5 p.m. So we'll be coming up with few more sessions. Few people have already agreed. So that's how it is going to happen. So thank you so much. I think most of the people are more looking forward to systems pharmacology. Of course, there are anyway, the session is happening on 12 p.m. at April 14th on COVID also. So looking forward to both the sessions. Hopefully, I know that you have only option to choose only one. Unfortunately, sorry, I should have made an arrangement that you can choose both. Anyway, so thank you so much for your time. It was really a bit lengthy, and it is already 6:35. We have spent almost 1.5 hours on QSAR. I hope it was helpful, and you will try to come up with something more reproducible and reliable QSAR models for the chemist and experimentalist to work on, so that your models will be reused by many people, so that rather than a publication, it will be on table. You can make a really good molecules based on your models. Good luck, and thank you so much for your time and your coordination. And take care, and stay healthy. Bye for now. Thank you.